Molecular structure of DNA and RNA. Prepared by Maria Luisa A. Monsanto STEM 11A. DNA discovery. DNA was first observed by a German biochemist named Friedrich Miescher in 1869. But for many years, researchers did not realize the importance of this molecule. Friedrich Miescher. It was not until 1953 that James Watson, Francis Crick, Maurice Wilkins figured out the structure of DNA, a double helix, which they realized could carry biological information. Maurice Wilkins, James Watson, Francis Crick. What is DNA and RNA? DNA stands for deoxyribonucleic acid. It is a nucleic acid that contains the genetic instructions used in the development and functioning of all modern living organisms. The blueprint of biological guidelines that a living organism must follow to exist and remain functional. Nucleic acid. Nucleic acids are molecules that allow organisms to transfer genetic information from one generation to the next. Nucleic acids are composed of nucleotide monomers linked together. RNA stands for ribonucleic acid, one of the cell's key regulatory players, where they catalyze biological reaction, control and modulate gene expression, sensing and communicating responses to cellular signals, etc. Transfers genetic code needed for the creation of proteins from the nucleus to the ribosome. Three main types of RNA. Messenger RNA, mRNA, transcribes genetic information from the DNA found in a cell's nucleus and then carries this information to the cell cytoplasm and ribosome. Transfer RNA, RNA, is found in a cell cytoplasm and is closely related to mRNA as its helper. RNA literally transfers amino acids, the core components of proteins, to the mRNA in a ribosome. Ribosomal RNA, RNA, is found in a cell cytoplasm and is closely related to mRNA as its helper. RNA literally transfers amino acids, the core components of proteins, to the mRNA in a ribosome. Structure of DNA and RNA. Structure of nucleic acids. Each nucleotide is made up of three parts: nitrogenous base, five-carbon sugar, phosphate group. Phosphate group. Phosphate group is an important structural component of nucleotide, which is the basic structural unit of DNA and RNA. It is a component of energy-rich molecules such as ATP. It is also bound to coenzymes like NADP and ADPH involved in anabolic. It is also a part of the hydrophilic head of phospholipids in biological membrane. Sugar. Sugar forms the central molecule in a nucleotide. 5-carbon sugar forms a nucleic acid. Two types of 5-carbon sugar: deoxyribose, DNA, ribose, RNA. Nitrogenous base. A nitrogenous base is an organic molecule containing both carbon and nitrogen atoms. The name nitrogenous base signifies that several nitrogen atoms act as bases in solution. In nucleic acids, nitrogenous bases contain either one ring or two fused rings. Purines, double ring. Pyrimidines, single ring. DNA structure. Double-stranded. It has two nucleotide strands which consists of its phosphate group, 5-carbon sugar, the stable 2-deoxyribose, and 4 nitrogen-containing nucleobases, adenine, thymine, cytosine, and guanine. RNA structure. Single-stranded. Composed of phosphate group, 5-carbon sugar, the less stable ribose, and 4 nitrogen-containing nucleobases, adenine, uracil, not thymine, guanine, and cytosine. Stability of DNA and RNA. Deoxyribose sugar in DNA is less reactive because of CH bonds. Stable in alkaline conditions. DNA has smaller grooves, which makes it harder for enzymes to attack. Ribose sugar is more reactive because of CO, hydroxyl, bonds. Not stable in alkaline conditions. RNA has larger grooves, which makes it easier to be attacked by enzymes.